Hi Year 12s. Today we're going to be looking at factors that affect metabolism. This is our second video on the metabolism topic. In our last video we looked at what metabolism was and how different chemical reactions worked in different metabolic pathways and the advantages and disadvantages of using metabolic pathways. In this video we're looking at the external and internal factors that can affect metabolism. Let's begin. First off, we're going to talk about environmental factors that can influence metabolic pathways, such as temperature, pH, cofactors, and inhibitors. With temperature, generally the higher the temperature is that the cell is exposed to, the more energy that's available inside that cell, and the faster the molecules can move. The more they move, the more they collide, and the more chance of reaction taking place. The detrimental side of this, as we know, is if you reach temperatures that are too high, you'll exceed the optimum temperature that the enzymes work best at, and if you increase the temperature too high, you'll actually get the enzymes denaturing and the reaction rate decreasing at a quite high level. Same thing with pH. All cells have optimum pH that they work best at. If you go too high, those enzymes will denature and the chemical reactions and metabolic pathways just won't work. Cofactors. Cofactors are special chemicals that bind to the active site of enzymes. They can be organic or non-organic. Organic ones are known as coenzymes, which are really important to the cell. Basically, the purpose of a cofactor is to help the enzymes work more efficiently. Lastly, we have inhibitors. So the presence of chemical inhibitors prevent enzymes from performing their function. So obviously there's times when we want an inhibitor, such as some antibiotics blocking the enzymes in viruses from working, allowing us to defeat the virus or the bacteria. And sometimes obviously they're bad because they're stopping the chemical processes in our own bodies. There are many chemicals that we use in our everyday life that actually can interfere with cell metabolism. The way they work is they alter the structure or function of an individual amino acid, and that amino acid could potentially change the shape and function of the entire enzyme. Many of the antibiotics and drugs that we use are considered to be inhibitors, so there has to be a lot of research into those before they're released widely to the public to ensure they're fit for medical purposes and not going to interfere with our cell metabolism in a detrimental way. We're going to look at 4 different types of chemicals that are used in everyday life to have a look at how they can interfere with cell metabolism. The first ones we're going to look at are poisons. So poisons work by inhibiting enzymes involved in metabolic pathways. There's 2 main poisons that we're going to talk about. We're going to talk about cyanide and malonate. So cyanide, or otherwise known as potassium cyanide, is an irreversible inhibitor of an enzyme in the electron transport chain of aerobic respiration. Therefore, if it's blocking an enzyme in the electron transport chain, it's limiting the amount of ATP production that's going to be produced in the mitochondria. If you're limiting the amount of ATP production, the entire cell runs out of energy and causes death. If that's affecting enough cells, the whole body's going to run out of energy and ATP, and that can lead to the entire organism's death. Same with malonate. Malonate is a poison that binds to the active site in an enzyme that's essential for the Krebs cycle and the electron transport chain. So this pretty much stops all ATP production, and usually any exposure to malonate generally results in the organism death. Herbicides. Herbicides are chemicals that we use to kill or greatly slow the growth of a plant. Very simply, the way that these work is they act as competitive inhibitors, and they'll bind to the active site of an enzyme that's going to help the growth of the plant. If you block the active site of that enzyme that helps grow plants, obviously the enzyme can't work and the plant can't grow. Medicines. So medicines that we use in everyday life generally act as inhibitors to viruses. So not our bodies, to our viruses. So one of these inhibitors can actually affect the enzyme protease, and protease helps to build up proteins. If we're stopping the enzyme protease from working in viruses, the viruses can't actually grow and build new protein coats, which means they can't disperse throughout the body, stopping the entire transmission of the virus. Penicillin is another medicine that we use quite frequently, and that works by inhibiting enzymes building cell walls, and we know that cell walls are really important inside of bacteria, viruses, etc. Antibiotics are really useful to us as a population because they're helping us kill pathogenic bacteria. But obviously their overuse has also detrimental effect. Their overuse has led to the rise of superbugs, and now we have bacteria, etc., that just aren't responding to antibiotics, which is a detrimental effect of the use of medicines. Lastly, looking at insecticides. So insecticides are chemicals that we use to kill insects and pests. They're beneficial because if we're killing off insects and pests that are eating all our crop, obviously it's going to improve the yields of crop, meaning we get more crop in the long run. We use both naturally made and artificially made insecticides. One of these that we're going to look at, which obviously had the useful fact of improving crop yields, was DDT. And you've looked at DDT since about year 9 in every science topic you've done. DDT was developed in 1944, and the way it worked was to disrupt the nerve impulses in insects by opening the sodium channels and just flooding their nerve cells with basically electrical messages. That was fine because we were only using amounts of DDT that were harmful to insects. There was no risk of it being harmful amounts to other animals or to humans. That being said, DDT is banned today because what they didn't do is research into the fact that DDT was actually an accumulative poison, which means it doesn't break down in the body. It just builds up. So the more of those crop you ate, the more of the DDT you had in your body. The more DDT you had in your body, the more chance of becoming toxic. We stopped using it when we found that it was present in toxic amounts inside of humans.